Bonjour tout le monde. Nicolas Chaumont et moi-même sommes les coprésidents de la session Sciences fondamentales et nous avons le grand honneur de présenter aujourd'hui Dr Ben Berkhout pour notre séance plénière en sciences fondamentales. Et il est arrivé hier soir d'Amsterdam. So it's a great pleasure to introduce Dr. Ben Berkhout for the keynote speaker in basic science. So Ben studied molecular biology at Leiden University in the Netherlands, and he received his PhD in 1986 on the regulation of gene expression in bacteriophage. So he then went to the United States for 5 years as a postdoc, and he started at Harvard Medical School in immunology. He then joined the NIH in 1988, and this is where I met him. We were postdoc at the same time, working under the late Katie Zheng. This is where he started to work on HIV. Because he was fascinated by RNA structures, he started to work on the TAR RNA, and I remember when he was working on what he called the nascent RNA, the target of TAT, that was published in Cell in 1989. He was taking RNA structure and was folding them on paper over and over again before starting an experiment, because at that time he didn't like computers. But when his idea was finalized, he did not stop the experiment until his model was demonstrated. That's a key feature of Ben. When he starts, he doesn't stop. In 1991, he returned to the Netherlands, and he has been there since then. He's at the University of Amsterdam as a professor of virology since 2003, and now he's the head of the experimental virology lab. He has been very productive with more than 500 papers on a wide range of topics, most dealing with HIV, going from reverse transcription to drug resistance, and from translation to entry inhibitors. His special interests remain in viral RNA molecules and virus evolution. He has trained more than 45 PhD students, and he's very active as an editor. He's the editor-in-chief of virus research and the editor of several journals. He has been on many international science committees, and he received many awards, including the Retrovirology Prize in 2008. So after attacking the viral HIV RNA for gene therapy purposes, he's now taking scissors to cut the HIV viral DNA, and this is what he's going to talk about today.
Okay. Now, I was asked to start by declaring that I have no conflicts of interest. And then the second important statement, of course, will be that I will try, this time, to stay in time, despite what Anne just said. And thank you for the kind words of introduction, Anne and Nicholas. Thank you for the invitation. It's always good to be back in Montreal, a main hub for HIV-1 research, and especially also, for me, the molecular side of things. What I'm going to present today is, I guess, a few translational stories. So it will be laboratory research, molecular virology that we do, but it has a purpose. We try to develop either new therapies or new diagnostics and so on. And I guess if you're and you're thinking, okay, is there a common theme in this presentation? Then I'll help you. There is one, and that has to do with virus evolution. Now, as I know for clinicians, if HIV-1 evolves, it's usually bad news because the virus becomes resistant. Now, for basic scientists, it actually sometimes it's good news in the sense that it's the start of doing interesting stuff, finding out how resistance mechanisms work out, and actually by doing so we try to be in communication with the virus, doing evolution studies to learn new mechanisms that then later on we can use for applications. Okay, let's see if this works. So this is our playing field, not a patient, but it's a T cell that can be infected. And I guess I just quickly go through the cycle and point out a few points that will become important later on. So of course the virus has an RNA genome which is reverse transcribed. It's converted into double-stranded DNA that then is imported into the nucleus, and that's a unique property of this type of virus, where it's integrated into the human genome. And then we have the cycle, and of course then you get new RNA, new protein, and new viruses being produced. The cycle takes 1.5 days, one virus gets in, and 1.5 days later, thousands of new viruses are produced. So it's quite an amazing amplification machinery. And I indicated the 4 inhibitors that you all are aware of that have different points of attack. Now, if you're going to talk about CRISPR-Cas attack, of course, we are going to look at the DNA form here or there. And of course, this integration of HRV1 makes it a very difficult target for cure attempts. Hep C virus is easy, relatively easy to get a cure if you have the good drugs. Many other viruses are relatively easy to cure, or they are cleared spontaneously. HRV1 is quite a different story, and a big part of the answer is because of this integration into our genome. So this is the picture you're probably also all familiar with. We can effectively treat patients, but we cannot cure them. So that's shown here. There is very effective control of the virus, and that can be for many, many years. But if the therapy is stopped, soon the virus will rebound. And that made us think about alternative strategies. And the clinical study that I found very decisive in our future, in defining our future research, and that happened about 5 years ago, was the so-called Visconti trial, where they started treating patients early for 2 to 3 or 4 years. And then, not all, but some of them actually, after that initial treatment period, were able to control the virus. So there is some control possible, and we just have to, perhaps by gene therapy, manipulate the system a little bit to get this balance in the right direction. Now, we think about a gene therapy because we would like to, unlike the regular therapy, we would like to have a long-lasting therapeutic effect after a one-time treatment. Yeah, that's the idea. It's a long route, but that's the idea. So the tools that we're going to use for this is a viral vector to deliver the gene therapy. That will be a lentiviral vector. We purposely choose this vector because it's based on HRV1, and it also integrates, and you need that integration to get a durable effect. The target cells we have, I'll show in the next slide, are hematopoietic precursor cells, and if you modify these cells, then all the progeny, like T cells of course, but also monocyte and dendritic cells should be modified and hopefully protected against HRV1. I will talk a little bit about RNA interference and CRISPR-Cas in the next slides. And then the idea is that these resistant cells will preferentially survive, and they should lead to a partial repopulation of the immune system. So this, and after this slide, I think we move into the lab. So this is sort of the picture we have. This will be a patient infected, treated for a long time, and likely failing on the regular therapy, and then he will be offered a gene therapy, which starts by a pre-treatment with GCSF to mobilize the hematopoietic, the blood stem cells from the bone marrow into the periphery. Then from the periphery, we will collect the stem cells, the CD34 positive cells. They will be transduced with the lentiviral vector ex vivo, in the test tube. And then the next day, these cells can be reinfused back into the same patient. The modified cells should preferentially survive from that point on. And again, if this is going to work, it will be a one-time treatment. So here I indicated the points of attack of RNAi and CRISPR-Cas. RNAi is attacking RNA molecules, and if you want to attack HRV1 RNA, then the targets are here. We know that this is actually not a good target because it's not a naked RNA, but is protected by the nucleocapsid protein. That's why attack doesn't work here, but of course newly made RNAs are vulnerable from attack by RNAi. CRISPR-Cas is attacking DNA, double-stranded DNA, so we have to look at the target here, unintegrated DNA, and then upon transfer into the nucleus and integration, it will be the integrated provirus. That's the point of attack. Now, if this DNA is attacked, it probably will be degraded. If this DNA is attacked and cleaved, yeah, it's one of our chromosomes, then of course, very quickly, a DNA repair machinery will kick in to link the two pieces of DNA together again. And that will become important in the following part of my presentation. So here I listed a few of the essentials of, on the left-hand side, RNAi mechanism, on the right-hand side, CRISPR-Cas. It's not all that important. I just focus on a few points. As I said, one attacks RNA, the other DNA. But they are very similar in the sense that both mechanisms are exquisitely sequence-specific. Yeah, and that's what we like in a therapy because we would like to target HRV1 only and not cellular RNA, cellular DNA that's around. It's also important to realize that actually RNAi is more simple because you only have to introduce a so-called short hairpin RNA in a cell. The cell itself, all our cells, they have the RNAi machinery. It's used for human gene expression regulation. CRISPR-Cas is more difficult because it's of bacterial origin and you have to introduce all the components. And that means you have to bring in an enzyme and you have to bring in a guide molecule that guides the enzyme to the DNA for a double-strand DNA cleavage, and here the RNA will also be cleaved. Now I just, here on the lower left corner, I summarized, I think, more than 10 years of research we did on RNAi. I do that on purpose because I initially thought, well, with CRISPR-Cas, that's nice, new tool, but probably we will find very similar things. And I was much surprised that actually every experiment we did, we got a quite different result. So if you attack RNA, if you attack HRV1 with RNAi, our initial observation was that you really have to work hard. You have to test many short hairpin RNAs to deliver the attack, because many of the short hairpin RNAs are not active. We tested at least 30 or 40 to come up with a handful of potent inhibitors. Then we got very good inhibition, yeah, that made us working on this much further, but eventually the virus always escaped. And that happened within weeks, many weeks, or a month. The way the virus escaped was by selecting simple point mutations in the 20 nucleotide sequence that was targeted. And these point mutations could happen anywhere in that window of 20 nucleotides. Of course, this demonstrates how sequence-specific the attack is, but it also tells you how easy it is for the virus to escape. Now, the lesson that we learned from the clinic and the use of drugs is that if you want to prevent viral escape, that lesson also holds here. You have to think about combinatorial approaches to prevent the virus from escaping, and that worked. So let's now move to CRISPR-Cas. When we started around 3 years ago using CRISPR-Cas, we were certainly not the first one starting to use it against HRV1. We're probably one of the last groups, I would say. But there were still very many question marks. And the one that I found intriguing is that several initial reports said that there is no escape possible if you protect cells against HRV1 using the CRISPR-Cas machinery. I thought, well, that sounds like impossible, and you will see escape will occur. Of course, CRISPR-Cas compared to RNAi has one additional interesting option. If you go for the DNA, you perhaps can attack the integrated provirus and start removing or mutating it as the source of the reservoir that you want to attack in a cure attempt. One slide, one result from the RNAi experiments. So here we are targeting HRV1 in a sequence in the genome here, which is encoding the protease. Good inhibition, really good inhibition, but the virus always escapes. And if you look in different cultures, you will see that the virus picks up a point mutation. If you look more carefully, you see that actually the virus is selecting certain point mutation because the virus selects only point mutations that are so-called silent codon changes. So the sequence is changed, but the encoded amino acids of the protease protein are not changed. Yeah? Of course, all the mutations are made, but these are the only ones that HRV1 allows for the escape against the therapy. Okay, CRISPR-Cas. You know, one more slide, probably you see it in your favorite newspaper, CRISPR-Cas and so on. So this is how the molecule schematically looks like. It's of bacterial origin. It encodes the Cas9, which is a double-strand DNA endonuclease. Yeah, that's going to trigger the cleavage. And the process is guided by an RNA guide, which is indicated here, that forms nice base pairs with a piece of DNA, double-stranded DNA, which forms a bubble, and then you get DNA cleavage on both strands. Okay, so our first experiment, based on the RNAi expertise, we thought, well, we better start testing a lot of guide RNAs against HRV1, because perhaps only a few of them are active. So that's why you see all the arrows here on top. At all these positions, we started attacking the HRV1 genome, and we initially tested the activity of these systems in a very simple transient assay system. For those of you who are interested, regular 293 T cells, in which we transfect all the components, Cas9, the guide RNA, or the gene encoding the guide RNA, and the gene, and of course HRV1 to score virus inhibition. Now all the controls, 3 of them are on the left-hand side. There you score high virus production, capsid P24, after 2 days after transfection. And then on the right-hand side, you see that actually all the guide RNAs we tested are pretty active. Perhaps one is more active than the other, but this is quite different from the RNAi experiments we did before. So apparently it doesn't matter that much which guide RNAs you select, at least for inhibition, because they all seem to work. That was the first surprise. The second surprise came very rapidly. We introduced the components into T cells, and we started doing HRV1 replication studies, and we got very good inhibition, yeah, very good inhibition. But for some of the inhibitors, within a week, we observed massive virus escape. For some, not for all, but for some. Very rapid escape, much faster than we have ever observed using RNAi and so on. So that made me intrigued about what was happening here. And the story became even more puzzling, at least for some time, when we started sequencing the escape viruses. Because I don't know if you can see it, but here are some of the HRV1 sequences that we attacked. And underneath there are the sequences we see in escape viruses. And perhaps you notice already that it looks quite unusual, because at least for some of them, you don't see point mutations, but you always see small deletions, especially deletions, or small insertions, indels, as we call them. And I think, I also cannot see it, but I think they're, for instance, here and there. For others, you don't see the indels, you see smaller mutations, but these are probably, again, highly conserved sequences. Here we are actually in the LTR, very next to the Tata box. Very important sequences where the virus probably doesn't allow indels to be selected. So indels, very rapid escape. What's happening here? And there's another strange thing. Oops. Can we move one slide forward? Because when we looked at all these cultures, and the replication curves are up there, we actually noticed that the events can be split in 2 groups. On the one hand, we have seen the indels here for this group, when escape occurs very rapidly, and that happens only for the non-conserved viral targets. If you target viral sequences that are more conserved, it takes much longer for the virus to escape, and there you see the more classical pattern of escape by the selection of point mutations. Okay. It even became a little bit more bizarre, but this also, for me at least, gave the answer to this puzzle. Because all the mutations, all the indels, and actually also the point mutations I just mentioned, they all seem to cluster exactly around the position of DNA cleavage. Whereas with RNAi, I told you, the mutations can be selected in the complete window of 20 nucleotides. Here, everything is clustered around the point of attack, as if the mutation is sort of linked to the event of DNA cleavage. And that is indeed the case. And actually, you know, I didn't have to think about it too long, because if you study CRISPR-Cas literature, you know that if our chromosomes are cleaved by CRISPR-Cas, that they will be repaired. The most popular, and also especially in the cells we use, repair machinery is called NHAJ, non-homologous end joining repair, and that repairs the DNA, but in the act, it actually introduces indels. So what's happening is, with CRISPR-Cas, we cleave the DNA, the cellular machinery kicks in to repair the DNA, introduces indels, and the virus picks those indels, at least some of them, for escape. Yeah, so the cell, in a sense, helps the virus to escape. And I thought that was sort of an interesting, unexpected twist. And here in the slide, I call that a yin-yang effect. Okay, virus escapes. Next step, obvious next step, is to think of a combinatorial approach. So we had all these inhibitors, so we started making many combinations, and I think we have around 10 combinations or so in total, and they are either based on GLTR7 with a second one, and I think 7 is there, yep, or it's based on GAG1, which is here, and then in combination with others, yeah? 10 combinations of 2 guide RNAs for a combined attack. The one that is marked in a certain color, but I'm a little colorblind, so I guess this, what is this, bluish, or? That's the best combination, but you know, you will see in a few slides. But there's nothing special about this combination in terms of position in the genome and so on. But that's going to be the best one. So we first, again, tested these combinations in the transient assay, simply scoring virus production. And what we usually see is that, so it's listed as single combi, and so on. In general, the combination is at least as good as the best single guide RNA. So that makes sense, nothing special. Then we did a lot of virus cultures, yeah, to study virus escape. And I guess I have to explain this slide a little bit, because what we score here on the axis, a little bit unusual, is the day of virus breakthrough. So we simply cultured, we looked, we always had initial inhibition, but then we looked whether the virus escaped. And here are some controls, but here, for instance, these combinations, we get escape within 1 or 2 weeks. Yeah? If you sequence the escape viruses, again, the same story, you see the hallmark, the signature of NHAJ-mediated DNA repair, small indels. Okay, here it gets a little bit better. You have to wait 1 or 2 months, but then the virus escapes. Yeah, and this is what I plotted here as the average of 4 cultures. Of course, virus evolution is a chance process, but you see, you know, there are decent small bars, error bars on these bars. And then we go to the 2 combinations that are the best. That is GAG-TAT-REF and GAG-envelope. Because for the duration of the experiment, which took around, 4 months or so, and then we stopped it, the virus did not escape against these 2 combinations. Yeah? Both combinations tested in 4 independent cultures. Then we thought, hey, this is interesting. So, we can inhibit this virus. That's not unusual. With these two combinations, we can prevent viral escape. So what's now happening in these cultures? Because, there's CRISPR-Cas all the time, and is it working on the DNA, and what's happening there? So we started analyzing these two cultures in very much detail. And we did two assays, a genotype assay and a phenotype assay. Genotype simply meaning that we're going to sequence the virus to see if we see the action of CRISPR-Cas attack. Then probably even more important is whether there still is some infectious virus present in these cultures. And we are going to do that by simple taking an aliquot and transferring that into a culture of unprotected, normal T cells, actually a T cell line that we use, just to score if there's any infectious virus left. So this shows the results of the genotype analysis, and so we have the two combinations, and we either look at the one or the other target, and we look early or late. And if you look at the numbers, you appreciate that early on a lot of wild-type sequences, wild-type HIV1 sequences are present, of course, yeah. We started the experiment with a lot of wild-type virus, but over time you see that the numbers come down. So wild-type virus in sequence analysis certainly goes down, and it goes down quicker especially for some of the guide RNAs. Now we also looked a little bit different because we also over time observed a shift from minor to major mutations. That's indicated here. So here we have the block of wild-type viruses that were there early, but not, or at least very little, at a later time point. But we also see that initially there were smaller mutations, 1 to 2 nucleotide point mutations, and they also disappear over time. And that's interesting, because that probably indicates, and that's consistent with literature, that if you have a target for CRISPR-Cas with 1 point mutation, that it can be re-attacked and re-cleaved. Yeah? So the cycle continues, and at the end, you introduce more cross mutations. Okay, now perhaps the more important test. Does this correlate with the loss of infectious virus? Well, indeed it does. So again, the two combinations here, 4 cultures, and we probed them early and at 4 time points. And you see initially, of course, there is wild-type virus there, because we did put in a lot of wild-type virus at the start of the experiment. That's not the point. But, and it happens quickly here, and it happens more slowly here, over time we start to lose infectious virus, to the point that we cannot get any infectious virus out of these cultures anymore. And we tried very hard. And of course, when you compare the two combinations, this one seems to be stronger than this one. And there could be many reasons for that. So that made us conclude, also in the paper that we just published, that we, and much to my surprise, I must add, that we are able to sterilize infected cultures using CRISPR-Cas. I find it quite amazing. Now, another thing that I should point to is that actually people, of course, started earlier than we on using CRISPR-Cas. The strategy they usually discussed, as the theory how things should happen, is that with a combination you will get cleavage here and there, and then actually you will remove the intervening sequences and simply truncate the provirus. Or even more simple, that could even happen with a guide RNA that looks at the LTR, because the second copy is there, so you cut at 2 positions and you remove the integrated proviruses. Sounds great. It does happen. If we do specific PCRs to detect such events, we will see them. They are there. It happens. But it happens very infrequently. And the major route that we describe is that nevertheless you can sterilize the culture, by virus inactivation. And that's shown here. You have the cleavage and you have the NHAJ repair introducing a red symbol which indicates the indels. If it's a minor indel, there will be recleavage, and again, introduction of indels that are more major, and by the end, the virus is not able to replicate anymore. So it's a proof of concept, this eradication, that it works in vitro. And I already, I guess, showed many questions are left. Why is this one inhibitor the best? And I listed some possibilities here. And it's also clear that much research is ahead of us. Do we have to work on the triple combination? What about the safety? Always a big question using CRISPR-Cas, especially in a gene therapy that will be on for the rest of the patient's life. Yeah, serious question. Does this work against other virus isolates? We have some initial data there. As we expected, it works against many other virus isolates and also different subtypes. We have to test this in other cell types, and we want to move that into a humanized immune system mouse model that we have operational. Now, so that's some good news, because a few years ago, I, of course, was quite pessimistic about the possibilities of a cure. I thought it was simply impossible for at least two reasons that I listed on this slide. One is that perhaps the gene therapy has to reach all infected cells, yeah? And words as all and complete are sort of, I find them non-scientific words because it's unrealistic. On the other hand, perhaps a sterilizing cure is perhaps not the only success we can go for, because a functional cure where we control the virus to some extent and then the human body, the immune system, takes over the control is something that's possible. So perhaps we don't have to reach 100% of the infected cells. And of course, certainly we have to improve vector systems, gene therapy vector systems, to get efficient in vivo targeting. That's a major bottleneck. And then a few years ago, I also thought that the inactivation of all integrated proviruses would be totally impossible. But we get close to that, and I realized immediately it's in a very simple laboratory assay with a T-cell line and so on, in a test tube and so on. But it's a proof of principle that actually, if you let CRISPR-Cas system go, it will sort of eat up all your infectious virus. And I think that probably, I'm not sure, but I think that probably many groups were on sort of the same track, at least with this exotic escape. Because soon after we published our work, many other peoples from different parts of the world appeared where they described very similar escape patterns. And actually the one that was very close on our heels was a group from here, local group from Montreal, Chen Liang, from the Lady Davis, who had beautiful deep sequencing results that confirm some of the things I already showed you. And then we wrote a very nice review together. But very similar results from Ohio, Kyoto, Utrecht, and I guess hopefully now we are again ahead of the pack by going into this eradication, sterilization scheme. Okay. There are some more minutes left, so I will have a short story on a different subject. It has to do with antiviral peptides. Probably you haven't heard much about antiviral peptides. Once they were popular in HIV field, it sort of has died out. And I guess for a good reason. The rise and the fall of T20, a beautiful new drug target, effective in patients, but the peptide has to be injected. And that is, of course, that is not the case for all the other effective drugs, and that's why it's not so popular anymore. But for us basic scientists, it's an intriguing molecule, new target and new resistance, and also drug dependence mechanisms that we are fond of understanding. And I'll show you a nice example where that can lead to. So this summarizes the 3 generations, as we call them, of peptides, not generated by us, but made by the industry, Trimeris, and then later on Roche. And you see here in this inhibition assay, you see the activity of the 3 generations, and you also realize how many peptides they tested, because T20 was one of the initial ones, but later on they were down to T2635. So that's impressive. But the improvement in activity is also quite visible. As I said, industry lost interest. Most people lost interest because of the injection issue and perhaps other issues. I don't know. But we, as basic scientists, we had already started working with these peptides, and we wanted, as Anne already indicated, we wanted to continue because there is more to gain, more to learn. And one of the first things we reported, which struck me, also compared to all the other resistance patterns that have been described, is that with the different classes of inhibitors, you actually get quite interesting differences in resistance pattern. And that's summarized here at the bottom. So if you go in with T20, in our case in cultures, but also in patients, you always select a mutation in the envelope gene, which is the target, actually in the GP41 domain, and usually it is V38 that mutates, mostly to A, but also to some other amino acids. Okay, then we came in with the 2nd generation peptide, and again we see the response at exactly the same amino acid residue. But what struck us is that you always get selection of a different amino acid. And here it seems as if you need a charged residue, all the charges are there, whereas here apparently it doesn't matter what's inserted, you always get resistance. And the 3rd generation is also quite intriguing because it is extremely difficult to get resistance. Eventually we got a low level of resistance, and I can tell more about that, but I think it's fair initially to say that there's no resistance, not like with the other ones. So can we understand that? Well, this slide summarizes a lot of work, but it tells you, I think, the complete story. So here on the left-hand side, we have the 3 generations of inhibitors. And basically what I indicated with the dashes, so this is the inhibitor, and this is the target, the GP41 protein. And you see more dashes, meaning there's a higher binding affinity of these peptides, 1st, 2nd, 3rd generation, for the inhibitor, correlating with increased antiviral activity. Then here I indicated in gray the mutations, and here are some molecular modeling pictures that give resistance. The type of mutations you select with T20 are in this box. Actually, these also give resistance if you test them, but they are usually not selected. So these are the mutations that either introduce a smaller amino acid, so you create a let's see, is it visible? Yeah, you create a hole here that disturbs the helix-helix contact, or you introduce a bulkier amino acid, for instance, an aromatic residue. And then you actually, you interfere with optimal helix-helix contact, and that's the mechanism of resistance. With the 2nd generation, that does not work, it's not sufficient, and you select, as I already said, charged residues. That's indicated here. You see a few charges, and either you get repulsion or attraction. It doesn't matter. Both are bad for the optimal helix-helix contact. And that provides resistance. And as I already indicated, no resistance initially for this compound. Now that, again, actually tells you an important story. And we did some more research. I skipped the results. But basically, one idea, if you look at these slides, is that over here, it may be possible that here this charge will not cause resistance because there is actually an opposing charge within the peptide. A salt bridge is generated there. That was not done by us, but that was done by Trimeris, and they did it to stabilize the alpha helical peptide, and it has much increased T-half values in blood and so on. It works. But it also provides an unexpected mechanism to avoid the development of resistance. because of the interactions within the peptide, it cannot be used for the virus to escape from by introduction of a charge in GP41. Okay, then a few more slides on the last bit. And again, you probably, after the first two sentences, you think, okay, where are they going? What does this mean? Because here we became interested in another peptide. I can immediately add a peptide that is much weaker than T20. So why bother about it? Well, we thought it was, again, interesting from a mechanistic point of view. Because it's a peptide, a ferripeptide, published by others, that is targeting the fusion peptide in the GP41 domain. And we thought that was an interesting novel target. We were able, not such a good inhibitor, but we were able to select decent VRIP resistance, and again, VRIP dependent viruses. And the critical thing here is that the mutations did not end up in the binding site of the peptide, because then there should have been, can we go, because then we should have expected mutations in the fusion peptide, which is modeled here. We never saw any mutations in the fusion peptide, probably because it's highly conserved and so on. But, of course, the virus was resistant, so we figured out where the mutations are that cause the resistance, and they are actually in the other domain, in GP120, in the C1 domain, which is not very close if you look at the structure model of envelope. So what are the mechanisms for the resistance, the dependence, and so on? So that's why we continued on this. And then we got some, and we are close to the end now of the presentation, we got some intriguing results. Because the mutations that we observed in C1, if you introduce them, in the test we did, if you introduce them in the wild-type protein, they stabilize the envelope protein greatly. That means the protein becomes more thermostable, and it's also less prone to CD4-induced decay. And it seems to freeze an early conformation of the envelope protein, and the dependence we explain by the peptide interacting with that protein and sort of acting as a wedge to induce the conformational change. But I would like to focus on this more stable envelope protein that came out of this peptide resistance study. And then I started talking with my close colleague, Roger Sanders. He's doing quite well in the HIV envelope vaccine field. So we are happy to have him close by. And of course, he was interested in this mutation because he said, well, we are, for the last 25 years, we are looking for mutations that can stabilize the native structure of the envelope protein. So let's test if your mutation is helping us out. And of course, I would not be telling this story if it would not have a positive outcome. So he tested some of these mutations, resistance, dependence mutations against ferripeptide, and he introduced them in envelope, and he did envelope and also vaccination studies. That's spelled out here, so this is sort of a few of the things he's interested in. You have to look at different envelope subtypes to broaden the response. He also wants to reduce flexibility in the protein, and of course you would like to mask domains like the V3 domain, which are very immunodominant, but they never induce neutralizing antibodies, and you want the immune system to focus on the more important part for raising antibodies. And he did studies in rabbits, and I'll end off with last slide showing a beautiful result because, and I'm a little, that's sort of a little bit what he told me, but this is a result that has not been reported for the last 20 years in HIV-1 research. So this is vaccination of rabbits with an envelope protein that forms non-native trimers, and you see it. If you do cryo-EM, you see, the protein going everywhere. Versus one that has, actually I must say, multiple mutations, but including this important C1 mutation to stabilize it. It shows much more discrete propeller-like trimer structure, and it is giving a response that leads to neutralization. And this initial thing was only done with an autologous virus, but as I said, this result has not been reported before in literature. So with that, I'd like to end. Hopefully I convince you that it's sometimes good, perhaps not always, but sometimes good to go in depth in mechanistic studies. Keep your eyes open, listen to the virus, trying to figure out what the virus is trying to tell you. Then describe, of course, all these nice mechanisms, but perhaps also do something unique with it. And of course, there were many people involved that did all the work. And here are I mentioned the RNAi work, but that's sort of the past. CRISPR-Cas is most work was done by PhD student Kang Wang under supervision of Ats Das. And I mentioned already the Roger Sanders team for the studies on T20 and VERIP, and then to the vaccine test that they performed. Thank you.
I think we are running a little bit late, but if there are some questions in the audience, yes? Does anybody has a question for Ben? I have one about the delivery of CRISPR. How do you think we can deliver CRISPR to be used for a cure?
Yeah, good point. Always delivery, at the end of the day. Yeah, well, the route for me will be simple, especially if you go for HIV-1. Again, we would like to use a lentiviral vector. And on paper, it looks simple. You insert the Cas, the endonuclease, the gene encoding the endonuclease, and the 2 guide RNA genes on that lentiviral vector. But in reality, that already is a major issue because of the length of the Cas enzyme and so on. So the titers of these factors drop. So there's a lot of technology development needed to make a smooth transition to the application.
So we thank Ben for his great presentation. And I think this is a coffee break now, and then you go to the other sessions. Thank you very much for listening.